I give you a short introduction on melanoma and then quickly change gears towards more single cell stuff. But as you probably know, let me see now if I can master this yet. Oh, yeah. As you probably know, melanoma originates from the malignant transformation of melanocytes, right? And melanocytes are the pigment-producing cells in our skin that reside at the basal membrane. And the way we think, this is a very oversimplified way of putting things, but it's a nice summary slide. The way we think is melanoma first through oncogenic hits in the MAP kinase pathway, majorly BRAF and NRAS mutations, also some NF1 mutations. But as we think of it, this is a multi-step process during which molecular alterations accumulate. So at the end, having these oncogenic hits is not enough to get a full-blown melanoma. There's many other molecular alterations that have to occur to give a full-blown melanoma. And the way we think melanoma is growing, and this is really textbook knowledge, is first it grows radially, then vertically, before basically hitting the next blood or lymphatic vessel and start spreading through metastatic dissemination. And once metastasized, it's really nasty. So if it's a primary tumor, you know, normally things are cut out and then you're fine. But once metastasized, melanoma is still a very, very deadly disease. What about treatment? I think we made a large progress. We came really a long way in the last 10 years with the onset of targeted therapies and checkpoint therapies. But what you can see from these slides, we clearly have to do better, since only about 30% of the patients, metastatic patients that are treated with targeted therapy, will meet the 5-year survival mark. And about 50% of immune checkpoint treated, and that is the combination of anti-PD-1 and anti-CTLA-4 treated metastatic melanoma patients, only 50% will reach the 5-year survival mark. So the majority is relapsing. And you may wonder why it's so difficult to treat melanoma. So what you have to keep in mind about this nasty cancer is it's the most mutated cancer amongst them all. And you know the reason for that, that's basically UV light, right? So if you were to order mutation load over cancers, you find melanoma on the very right-hand side here. So the most mutated cancer, but also the most genetically heterogeneous. So if you plot now numbers of different genetic clones within a single tumor, or tumors, you will see also here melanoma is on the very right-hand side. So genetically very heterogeneous, and on top of it, this cancer is very plastic, and leading to genetic, but more importantly, non-genetic resistance mechanism. When I talk about plasticity, what do I mean by that? We have another term for this plasticity. We also call this phenotype switching. What is phenotype switching? It is the melanoma cell's capability or capacity to switch back and forth between transcriptional programs. And this slide is actually it's historically grown. So initially this was identified by Keith Hook, he's from Australia, I think, but then in Switzerland. He published in 2008 a seminal work about identifying the first transcriptional signatures in melanoma, and he historically termed these two opposing transcriptional states as proliferative and invasive. And basically, the melanoma cells can switch back and forth between these programs through cues of the microenvironment or through, for example, treatment or drug pressure. And these signatures are by and large mutually exclusive, and we believe they're independent of any driver mutation status. So they historically, as I said, termed this proliferative invasive, but now you see that these transcriptional phenotypic states are also functionally rather opposed. And now we know, with the proliferative state, we have differentiated, sensitive, etc., and then the opposing functional characteristics on the other side. Just one more word about MITF. So one transcription program is called MITF high, the other one MITF low. For the rest of the talk, it's important to understand what is MITF. And MITF is the master lineage or master transcription factor for the melanocytic lineage. Full house. And so basically, it's very important to understand this transcription factor. So MITF basically, as I said, is is a lineage-specific transcription factor. And basically what happens if you invalidate MITF genetically in black 6 mice, they become white because they lack functional melanocytes. So this transcription factor is important for melanocyte, but also melanoma biology. And the way we think, this is just a cartoon, way too small, but it controls basically every cellular function in this lineage. And what's important in the following of the talk is understand it's important to understand how this transcription factor works, as we believe it works like a rheostat model, meaning depending on its activity, it can do different things in the melanoma cells. So if you have a medium towards high MITF activity transcription factor, the melanoma cell is happily proliferating, everything's fine. If you have too much, it's not good. If you have too little, it's also not good, depending if you're the melanoma cell or you want to get rid of it. This is just to show, initially there were two states. We revisited this model in 2019, and you can already see it's way more complicated than that. There are way more transcriptional states. And that leads me to an overarching question of my research. And that is, we ask ourselves, what is the intratumoral heterogeneity? How heterogeneous is melanoma in a treatment-naive setting, but also, once you challenge it with drugs? And why is this important? It's important because you can think about, you can ask fundamental questions, as in, do all cells equally contribute to tumor growth? Do all melanoma cells equally resist therapy resistance, or do all melanoma cells have the same capacity to spread, right? And if yes, we have to kill all of them. If not, probably we have to kill the right ones. And these states can inform each other. So basically, this is a bit the background, right? And you're also aware that melanoma cells do not grow in an isolated fashion, but more like in an ecosystem, very much like a mangrove ecosystem, any ecosystem to the left. You're very well aware of the fact that melanoma cells are not growing in a petri dish, but they communicate with each other, but also with the cells of the microenvironment. And that, of course, increases the heterogeneity, the intratumoral heterogeneity. And the cool thing is now, since the advent or since the technical development of single-cell genomics, we can now capture this intratumoral heterogeneity, I would say quite precisely. There's still a lot of limitations there, but we came a long way, I think. And even on different molecular levels, if we have the capacity, the power, the money, et cetera, et cetera. But, that's a way to get to the next level. And I think I almost feel like a kid at Christmas having a new microscope. With these new techniques, we can really, see things we haven't seen before. But, generating all this data, can this actually bring us forward in understanding better the disease and come up with therapeutic ideas? Let's see. But different molecular levels on the single cell level, we can also do this now spatially, and I will come to that later. And also there, it's quite impressive what computational biology can do, because we can now even start building predictions about intercellular cell-to-cell interactions. All this is great because it helps us with hypothesis testing, hypothesis building, testing, and gives us ideas where to start looking. And I think, now in the rest of the talk, I want to give you a couple of examples. That's basically the outline. Just a reminder, sources of intratumor heterogeneity, as I said before, are threefold actually. They can be cell autonomous, so intrinsic to the tumor cells, and on all different molecular levels. Sources of intratumor heterogeneity can be non-cell autonomous, meaning that the TME, the tumor microenvironment, dictates or the interacts with tumor cells and therefore dictates heterogeneity. And third, often neglected but just as important, are these stochastic sources. Okay, and less well understood but just as important, and this complicates things. But if you look in the when you look in the literature, by now, almost every cancer type has its own ecosystem, landscape, you name it. Most of them were historically built around single-cell RNA-seq initiatives, but now we are building on this. Now RNA, protein, spatial resolution, etc., etc. And you can see that one of the first ones, actually in 2016, was performed in melanoma. And this is the Tirosh paper in Science. That was great. That was a great paper, but at the time it was a bit too early. And what do I mean by that? So back then, they applied single cell in patient material in tumors from melanoma, and basically they identified two malignant states, two melanoma cell states. The ones I told you before, MITF high, MITF low, they called it MITF program and the AXL program. There was nothing, I wouldn't say nothing new there, but it was limited to two states. And when they looked at the microenvironment, okay, they could pick up a couple of immune cell types, but that was about it. I think nowadays we can do this way more precisely. Over time, it became clear, as I already said before, there must be more melanoma cell states. And there was a lot of work throughout these years in the literature, from bulk RNA-seq, the Solidal paper, Mathieu knows very well, single cell work on cell lines, we also contributed to that. Different animal models for melanoma, this was a separate fish from Richard Weiss' lab, already, proposed a neural crest cell state from in vivo material, a stress state, and again, a melanocytic or MRTF-like state. We also contributed with some work to this different melanoma cell states under drug pressure. And this was in 2018 when we used a PDX model, a BRAF mutant PDX model, let the tumor grow, and then started treating BRAF MEK, tumor went away, lay low, and come back. And we profiled this model longitudinally using single RNA sequencing and could identify drug tolerant states, as you can see here. They were interestingly all coinciding within the same tumor. Again, there was increased melanoma, transcriptional melanoma cell state heterogeneity, drug-induced. Now, this was 2018. Now, researchers look at this more systemically. And this is now work, some of this is preprints, but work from, again, Tiroche, Italianized group, and Benjamin Izar. He was also in the initial melanoma paper from Tiroche. Now, this is single cell work across 1,000 tumors, here spanning, 50 tumor types. And what they do is they identify, they call it gene modules, meta programs. Anyways, the idea is always transcription states that most likely confer a cell state, a cancer cell state. And when you look at this, that's quite interesting, these initiatives, and I think it's still pretty wild out there. At some point we have to think about consensus, but you can already see that a couple of states are repeatedly occurring, like there's several EMT states, stress states, cell cycle related state, and you name it. But yeah, as I said, it's a bit early days, but we take it from there. And but anyways, this wildness prompted us to build our own ecosystem, and we chose again melanoma in this case. And we wanted to do this in a cross-species fashion. Again, we started doing this with single-cell RNA sequencing, and it was a 10x platform at the time. But then we also built on this with single-cell latex sequencing, spatial approaches, and whatnot. So what we did here is in a treatment-naive fashion. So we wanted to see, what's the base level of heterogeneity in a treatment-naive fashion. We took about 20 whatnot metastatic lesions from human patients, BRAF NRAS mutated, whatnot, and we took 5 primary tumors from our GEM, genetically engineered melanoma model, and we took the NRAS infrared model here. And then basically we performed single-cell RNA sequencing. This is a dimension reduction plot, and we like to look at the ecosystem by dividing it in 3 compartments: the immune, malignant, the strong compartment. And then what we do is we subset each one of them, and perform higher clustering. Basically get higher resolution in the different cell types. That's the basics, and then we build on that. And I'm not going to go in detail, but you can compare mouse and human to quite some extent. There are definitely overlaps, also differences. I'm not going to focus on that today. There's also ways to probably access this data, and I think they're building at the moment a shiny app so you can look up your patches. Now what I want to do the following is I want to focus what we learned about this malignant compartment here in the mouse, and then what we learned about the conserved overlap between the 2 malignant compartments, and what we did so far. So this is the computational part. I also skip it now for time reasons. We can come back to this later. So again, the first thing I want to do is now I want to subset this malignant population. And as I said, we perform reclustering. And also, this is a busy slide. Only to show in a treatment-naive fashion, we were actually blown away when we looked at the heterogeneity. The heterogeneity in the treatment in this mouse model was way more pronounced than we thought before. Finding all these cell states, so these we termed these cell states, okay, and this was based on gene enrichment, different gene enrichment pipelines. But what we also like to do is using scenic, that's like transcription factor network inference on a single cell level, to basically identify the transcriptional programs, the transcription factors that keep in check or ensure these transcription programs and these identities. So in a treatment-naive situation, it was way more diverse than we thought. And the next question we asked was, is there any organization within these different cell states? on a different level. And what do I mean by that? It's rather slow. What do I mean by that is we took an algorithm, it's called cytotrace. It's a pipeline to assess basically gene expression diversity, with the assumption that a more undifferentiated, a cell with a higher potential still, like a more stem-like cell, has a bigger gene expression diversity, because the cell still can, become many other things. That's still a lot of fates. However, a cell that is committed, now like the melanoma cell, the melanocyte sitting in the skin, making pigment, has a low gene expression diversity because it's committed, it's only doing one job, doing pigment. That's the assumption under that, and we can apply this also to the single cell level. And weird, there's the color code is missing, but basically the more undifferentiated, meaning a high cytotrace score, is blue, and the less differentiated, the most committed is yellow. Okay, so when we looked at that, we saw a great range of different differentiation levels within our melanoma ecosystem. And then we also put this to the test by measuring the activity now of a differentiated, very committed melanocyte gene set, and this was very active here on the upper right. And you see this nicely correlates with this, also with this very general cytotrace method. And MITF follows that specific one. So that was interesting to see. So we asked ourselves, there's a lot of range. We also applied a lot of pseudotime inference techniques. I'm not going to show this since the reviewers did not like this at all. Anyways, but when we saw this, we were hypothesizing. So it looks like the melanoma cells can de-differentiate quite some. And the question was, if you were now to look at embryonic development as an example, how far can they de-differentiate back? And to answer this question, we turned to a study about neural crest, single-cell RNA profiling of neural crest development, because the melanocytes derive from the neural crest, right? This study here from Soldatov et al. 2019. They took a mouse embryo at E9.5 and performed also single-cell RNA sequencing, a lot of spatial. It's a beautiful paper, you can go to that. And what we did was basically, we took their information or their gene signatures, first of a very early, still, very, I would say, still very precursor-like neural crest population. It's called the pre-EMT population. The neural crest cells still reside within the neural tube. They're still very uncommitted. And this is all the data from Soldatov. This was here, basically, in a trajectory. This was the signature. And we took the signature and put this in our melanoma map. And interestingly, we were able to identify now a population down here in number 5 that was enriched for this signature. Hence, we call this population like the pre-EMT or more stem-like melanoma signature. Then we went down the road. So what happens next in embryonic development? These cells delaminate, start migrating before giving rise to different fates of the neural crest. And I think melanocytes is at E10.5 or E11. You don't see melanocytes here because it was too early. We took this intermediate states where the cells delaminate and start migrating. This signature was originally 0, also connected to 0 from 5 to 0. So this was for us a neural crest-like state. And then one state at the very bottom here, the very committed, is the mesenchymal state. Here, translated then here to this cluster on our melanoma map. So suggesting that the melanoma cells, can de-differentiate back all the way here to this pre-EMT-like state, and by hijacking basically the molecular logic underlying this neural crest cell state specification. So this link between development and cancer, is pretty, pretty obvious. Many people look at that or want to tie this back to embryonic development. It is not one-to-one, but you can see some remnants definitely of this. The remnants of these developmental programs, even in cancer. The next question we asked, so it's okay, that's nice, but can we learn actually new stuff, new biology from this? So also we were impressed or startled by the fact that this cluster 5 here, it almost looked like sitting on top of this hierarchy of differentiation. And so we hypothesized maybe this cluster 5 here is the one that is eternally fueling the tumor. And, eternally fueling a tumor, that's a cancer stem cell hallmark feature. And there you have to know that in melanoma, the last 20, 25 years, labs killed each other over the definition of a cancer stem cell in melanoma. Till today, it's not clear how melanoma grows. Is it hierarchical growth? Is it stochastic growth? No one knows by now. There was a lot of literature about ABCD5, really labs killing each other. And then there was silence because no one knew. There was a big, you know, years of silence. And anyway, so we said, okay, you know, maybe there's something special about this class of 5. Anyways, so we went in the wet lab, and now I will, you know, condense basically a Nature paper in a couple of slides, but bear with me. So Tuck is in our lab, very talented postdoc. Sorry, postdoc. What he did is try to, you know, at least assess this experimentally. What he did is he brought in a confetti allele on the NRAS background and the melanoma background. Basically, this allows, upon tamoxifen painting, this allows to turn on randomly colors colors in the melanoma now at a clonal resolution. So, you know, it's not like they all explode. This takes time, and it's really clonal resolution. He did a lot of 2D, 3D clonal analyses, proliferation kinetics, and whatnot. Then we teamed up with mathematicians because, you know, we are not good in this, coming up with a growth model, and this is what they pulled out with this double exponential fit. Basically, all this to say, yes, they found, or they could at least model, that melanoma would rather grow in a hierarchical fashion, where like a stem-like population has gives rise again to a stem cell and to a second progenitor P, which then gives only rise to P with fewer divisions. Okay, this was the mathematical data hinting towards. So now that, okay, that was nice to show, but you know, we wanted to actually tie this back now to our population 5 there, right? And the idea was, can we find these cells back? And can we actually inform now our melanoma landscape, our ecosystem? Can we actually take now an informed guess from this, right? This is what I said about hypothesis testing. It's not going to explain us everything, but it probably helps us with our thinking and building hypotheses. So what we did is we predicted cell-cell interactions, and we used NicheNet for that. There's different ones, there's Cell4DB, CellChat, and you name it now. This field is exploding. Anyhow, so what we did was using NicheNet, and this is based on known ligand-receptor interactions, and we were able to pull out a receptor that was rather specific for our cluster 5 here, which was Notch3. And Notch3 is known to bind a ligand which is called DLL4 here, and DLL4 now, going back to our ecosystem, was specifically expressed in endothelial cells. So, you know, this gave us an idea. Maybe these cells reside close to blood vessels, like in a perivascular niche or something, and thereby contributing to tumor growth. That was the idea. So then we went on and performed quite some spatial analysis, also because we had the luxury of having access to quite some cool platforms. I show you the less cool platform here. That's Vision. Probably heard this from 10x Genomics. This is not single cell resolution, clearly not. So what you have is you have these areas for capture areas on every area. This is what is it, 6.5 times 6.5 millimeter. You basically put your tumor section on top of it. You image this, you lyse your tumor, your RNAs descend on this support by gravity. And then in every little spot here, there's about 5,000 of these spots. There are 1 million probes mobilized. They have a poly T-tail, polyadenylate RNA binds to it. Below is a spatial barcode. You can cut it, you see DNA, you cut it off, and you can sequence. And then it's like a you have a spatial information with every transcript, and you can map it back spatially then. Cut a long story short, yeah, I said it's not single cell resolution. So we had, depending on where we were in the tissue, we had about between 10 and 20 cells within a spot. And also important to mention is there's quite some dead space in between as well, where you just cannot get the information. Anyways, what it does, it looks beautiful on very well organized tissue if you look at developmental, and beautiful papers with it. If you look at a heterogeneous tumor tissue, good luck. Anyways, what we did is then we took our single cell RNA signatures and put it back on and measured this. So now these are these pseudo spots now, and we can make this look nice without any distance in between, but there is this distance in between. Anyways, we were able to measure our pre-empty signature, and we found yes, and then we also colored our section by the distance to nearest blood vessel. We could actually show yes, it was localizing close to blood vessels, not to any blood vessel, but it was mostly immature blood vessels that were devoid of pericytes. So it was one technique we tried to hunt down these pre-empty cells. Next, we went actually to a cooler technique, I would say. It's called the stereo sequencing from BGI, and I think they just had for a couple of months had the Nature Cell cover with an embryo with this resolution. And they claim, this is a bit different technique-wise, this works with nanoballs, DNA nanoballs. They claim to have a 500 nanometer resolution, subcellular resolution. It's crazy. The thing is, if you look at the bin 1, which is the lowest resolution, 500 nanometers, you have no transcript. Okay, so you basically what you do is you accumulate the bins again until you have a signal. And then we came to a resolution of about 20 micrometer, which is close to single cell, still not single cell. And when you do that, you have about 1,000 transcripts at this resolution. So, but the cool thing is what we did then is, again, we took our signatures. And this is also nice because it's not single markers. You take 10, 20, 30, whatever, 40 genes. These are programs now, really. And when we did that, and for example, in the previous cell signature, you can actually see by eye, like something like a vessel structure appearing. That's pretty cool, right? And then, again, in the red dots, these are the pre-MP cells that we were interested in. It's very hard to see here. And then as a control, we took a cell state, which was a stress cell state, called the hypoxic cell state. So the geospatial profile was reminiscent of hypoxic cells. And then what we did is basically correlations, again, with the distance to the blood vessel. And yes, and this was very significant here, I mean, because we have, hundreds of thousands of cells. But basically, the pre-MP cells, the likelihood that the high pre-MP cell was way higher if you were close to the vessel, and it was the opposite for the hypoxic, high stress hypoxic cells that you had when you were far away from the vessels. Then we went to another spatial system, which is now not untargeted transcriptomics, but targeted, but really true single cell resolution. That's a very cool one. That's called molecular cartography. I think by now they roll out the machines as well. And there you can multiplex about 100 genes. And this is, if you want, multiplexed or, high dimensional RNA fish, if you want. And again, we took our pre-MP cell state, and now we designed for almost every cell state about 5, 6, 10 markers. And what you see here now, this is the segmented, representation already of our section. And now we have basically pseudo-coloring the cells according to our transcription profile. So now here, this is the accumulative signal, RNA fish signal of all these markers here. And then again, you can quantify, it's the same story again. Red is the pre-MP cells, closer to the vessel, and yellow is again the stress state far away from the vessel. And then here it's probably too small to read. This is then some examples of the single markers, and then here now every dot is basically a transcript. So computationally, I think it was also a bit of a new land. Also, when you talk to these platforms or these companies, they're on the computation side, they're quite limited at the moment. They wait for the scientific community to sort out, how to segment and using ML and these kind of things. So, but that's a pretty cool technique, and I think here we are true single cell. But then we did something everybody could do. It's we did just multiplex immunohistochemistry, and this was also a reviewer's comments I get. This is nice. Why didn't you just stay in notch 3 and Okay, yeah, we did that of course. And so SOX10 now, the melanoma marker and notch 3 was the highly expressed specific receptor I was talking about. In these pre-MP cells, you can see they nicely reside around these small but also big vessels. This is from the NRAS model. We had also similar pictures from the human. So yeah, this gave us a hint, it seems they're close to these blood vessels, perivascular lesions. That we wanted to learn a bit more about these pre-MP cells that we went in vitro. And then in vitro, basically, we did co-culture assays with NRAS melanoma cell lines and co-culturing this with BEN3 endothelial cells. And this co-culturing, significantly increases the sphere growth as such, but also the proliferative potential is shown by this watermelon system, where you can turn on M-cherry dox-inducibly, and then, if the cells proliferate rapidly, you dilute the signal, and you can measure this on the fax, and you see again, once you have co-cultured, you have a larger or significantly larger fraction of M-cherry low cells because they were proliferating more rapidly. And this is also to some extent notch 3 dependent. Also went in the mouse, again, melanoma cells labeled with red, the other one's green. The endothelial cells put this together, and almost in a dose-dependent fashion, you get an accelerated growth in the mice. In meat mice. And also there's stuff missing. So anyway, so yeah, this is what I made dose dependent. You know, this is 1-to-1. This is 1 time melanoma cells and 2 times endothelial cells, and 4 times endothelial cells connection. There's now a lot of follow-up work here to understand, you know, how is this working in terms of secretome and et cetera, et cetera. But I would stop here. Just to give you an idea, yes, we were able to, you know, get informed by looking at the ecosystem, and then finally, you know, pulling out with all this spatial stuff, pulling out basically a melanoma, I wouldn't say stemlet population, but a pre-MT state that is able to hierarchically fuel tumor growth. So that's a cool thing. And at the very end, I'm not sure if I'm over time, you tell me. Okay, so in the end, it will be the comparison between now the mouse and the human, and also again an application, what we learned from that. So again, we compared, we are also doing this now for the other populations, of course, but I want to stay with the malignant compartment. So we took basically the mouse and human, compared them, and in the end, we found overlap of about 6 to 7 evolutionary conserved melanoma cell states, as we call them. And this work is under revision at the moment. So there's something like, we call it the antigen-presenting melanoma cell state, the melanocytic we knew about. The mesenchymal-like cell state, this is probably going close to this undifferentiated or invasive-like cell state. We find a new request cell state, and this is all treatment naive. We find 2 stress states. I told you about this one before, and of course the pre-MT cell state. And the next question was, you know, what can we learn now from this evolutionary conserved state? And, you know, one big question everyone has in the field, and I guess here you as well, is what about treatment pressure, not targeted therapy, but now immune checkpoint therapy? You know, how would immune checkpoint pressure would remodel now the conserved melanoma ecosystem? And that's basically what we try to assess here in this prospective trial that was conducted in Leuven, where they basically profiled patients, about 20 patients again before treatment, and that's, you know, one part I showed you already before. And then after 1 round of anti-PD-1, right, it's like a 2 weeks window, very early on, they sampled the same tumors again. So now we have matched lesions before and on treatment to capture a very early response to anti-PD-1. And, you know, the whole thing I just talked you about, we did the whole thing again. And then see what happened. And also there, of course, then we were also rather lucky. I mean, still 20 patients is better than nothing, but it's not the world either. So statistically power speaking, we're still limited. We get probably first ideas, but, you know, this was also, you know, we had the reviews back now. This is one major comment as well, saying, you know, okay, nice, but, you know, still a small cohort. So yeah, it's always, the question is always, do you find a bigger cohort, or, you know, you have to find collaborators that have a similar cohort, and that's what we did basically. Anyways, but at least we have equal numbers about responders and 11 non-responders, what you would expect from a immune checkpoint blockade. So, long story short, from the 6 to 7 evolutionary conserved states, only 2 states actually were linked to outcome to anti-PD-1 after 2 weeks. I went the wrong way, I think. Yeah, and these are basic, just here, they were in bold. And this was the antigen-presenting cell state and the mesenchymal-like cell state. And also here, as you would probably expect, but this is now stratified before and on treatment, responders versus non-responders. This antigen-presenting melanoma cell state was associated with response to anti-PD-1, whereas the mesenchymal-like cell state was associated with non-response. These are the ROC curves. Again, I said, you know, yeah, I told you about the limitation. Also here, you know, this is a bit scrammed because of several outliers, but there is something there. At least, you know, I want to try to convince you that this mesenchymal cell state is really refractory to immune checkpoint blockade. This doesn't look like that normally. I will skip that because I want to focus on this mesenchymal-like refractory mesenchymal-like melanoma cell state. So this cell state, as I said before, historically, many people saw before, looking at bulk RNA sequencing data, also during targeted therapy times, this cell state was already linked to worse outcome to BRAF/MEK inhibition And you just showed me data, right? So I think, you know, this is well accepted that this cell state is refractory also to targeted therapy. And the problem we had, and this, I mean, the problem, what we saw is when we used now single cell resolution, we had a problem actually, because when we took now the 50 or the top 50 mesenchymal-like genes that were characteristically upregulated in this cell state, and now measured this on the whole ecosystem, what we mainly lit up was cancer-associated fibroblasts. Okay, so these melanoma cells, as again, de-differentiated and look almost like something else. This was a problem because, you know, now you can think about all the bulk data. So was this really melanoma cells, or where did your signal come from? So this was basically the conundrum we had. And then, and these are just a couple of examples. So Taiwan, Luncan, TCN, TCF4, all these mesenchymal-like genes were high in CAFs, but also in this mesenchymal-like population, and not in the other ones. So we basically then thought, okay, you know, looking only at mesenchymal-like gene doesn't work. So we have to find still some lineage identity, and the ones MITF-SOX10 everyone uses did not perform well. So we were, I'm not telling you now, but we were to find a way to tease out better melanoma lineage genes that were still present in this very undifferentiated melanoma cell state, which is the mesenchymal cell state. And these, for example, CDH19 and S100A1. You can see these are now almost absent in the CAFs, but strongly. And only the presence of both, you know, on the presence of both programs, what we call it now is a true mesenchymal-like cell state. Also, we went on try to validate this spatially, and here what we did is, I think it's also a cool thing because now, so this is based on the fusion of the Akoya platform that is antibody-based, so protein-based, with RNA-scope. Akoya will also is selling now the update, the fusion update, where they claim to do that. If you ask them how the technique, do it, then you would see. Anyways, we had a person in the lab that was hacking this herself, and you know, on the old version. And so basically, you can now have multiplex protein and multiplex RNA on the same section. And what we did here is, this is the Akoya picture, and there's only 3 cells now. These 3 mesenchymal-like cells. It's bad resolution, sorry. And circled here, these are the protein pictures, so they're high in TCF4. And then basically below, you again see the accumulated signal or now the RNA scope signatures, and these are the single ones. So the minimal lineage genes here, sorry, it's very slow. These are the single ones. And then the mesenchymal, this pseudo-colored, of course, the mesenchymal-like signature here, and these are the single ones. Again, it's both programs present in the same cells, right? Another way to look at this was we went back to our patients and performed normal Akoya or immunohistochemistry, TCF4 now again in white. And what we did then in the consecutive section, we stained for the BRAF V600E mutation. There's an antibody that is mutation specific, right? And you can clearly show these are melanoma cells being reached in this. And I think it's fair enough to assume that not every patient has this, right? I'm not saying, you know, we found the holy grail to resistance to immunotherapy, but patients having populations of these mesenchymal-like cells, cell states, you know, they will have a problem at some point that these cells being more resistant to anti-PD-1. Of course, we wanted to learn more about this mesenchymal-like, or this, what we call it now, the true mesenchymal-like phenotype. Again, Mathieu, I see you're writing transcription factors and regulons, right? So we inferred again transcriptional states and, transcriptional regulons and the transcription factors for all the states. And this is the mesenchymal-like cell state. The ones, you know, expected Cl1, twist1, I mean, this is known EMT regulons, transcription factors. And amongst those was also this marker I was talking about, TCF4. Again, just to show you again the selectivity on a single cell RNA-seq level, you know, this one devoid of SOX, this is where the TCF4 is always expressed. So this actually TCF4 we were crossing before, as I said, during targeted therapy times. When we back then still had this classification into invasive proliferative, which is now, you know, I think we know a bit better now, but nonetheless, what we did in a mesenchymal-like melanoma cell line in vitro, we knocked down TCF4 at the time, and we saw, okay, yeah, there was really, you know, matrigel invasion is greatly reduced. And then also the combination with the BRAF/MEK inhibitors using now an shRNA against TCF4 in this mesenchymal cell line resensitized to some extent the cells to even to target therapy. So what is TCF4? And there you have to be super careful in the literature because intelligently they gave two different genes the same genome. And you go actually to PubMed and they write with a big, exclamation mark, be careful. But basically, TCF7L2, it's another transcription factor that competes with LEF1 to binding to beta-catenin. It's also called TCF4. And you go to literature, there are even papers that talk about one gene and have tools of the other one and vice versa. It's a mess. Okay, this being said, we're looking at TCF4 that's also called ITF2 or E22. It's a BHLH transcription factor, recognizes E-boxes, is essential for plasmacytic dendritic cell development, known in brain development, and also schizophrenia, I think, and it's a known EMT regulator. Interestingly, we came over an old paper in mirroring melanocytes, where they still, this was 20 years ago by Vincent Hering's group, where they still called it ITF2 at the time. Interestingly, when they overexpressed ITF2 in these mirroring melanocytes, the melanocytic antigens that are normally recognized by the T cells were going down. So was MITF. That was interesting. So it's so slow. Okay, then we went to TCGA, just on the RNA level, on the bulk RNA level. We really could confirm that MITF and TCF4 is indeed very much added. What we did then is we took again this mesenchymal melanoma cell line and we knocked down TCF4, performed RNA sequencing. We were quite amazed to see that TCF4 seems to control different transcription profiles. So this heat map is basically just a selection of genes that go with these gene sets, but basically, as expected, when you knock down TCF4 in these mesenchymal cells, what you repress is basically, or what you lose, is this EMT hallmark invasive-like signature. I'm pretty sure sleep alpha 6 would be amongst those. But more things are happening. You see, if you knock down TCF4 in these mesenchymal cells, you somehow recover or you de-repress now genes or gene sets around antigen presentation and also interference signaling. And these are examples here. Almost as if TCF4 is like a brake on this immunogenicity of this cell state or cells being in the cell state. Also, when we did the opposite, a TCF4 low cell line that was high in MITF, we brought in carefully TCF4, because if you bring too much TCF4 in there, MITF goes down, the cells die. They don't like it at all. But yeah, so if you bring it in TCF4 carefully, MITF goes down, they're still alive to do RNA-seq, and what you see is now on the RNA-seq level, now the whole pigmentation signature basically goes away, and you start seeing or you start gaining this mesenchymal signature. So there's really something about this transcription factor, and pretty much in line with this suppressing immunogenicity. We could also show, with the help of the Passion group now in Essen, we could show in an autologous matched melanoma T cell system, which is called the MAML86 cell line. We also inducibly could then overexpress TCF4. We could see like an impairment, an inactivation of the T cells, as shown by the reduced reduction or reduced release of TNF alpha. We have also similar plots on interferon gamma. And just to show, this was the TCF4 overexpression, and then I think there's also stuff missing here. But basically, the overexpressed TCF4, MITF again is still going down, and this is on the RNA level. So we went on, and we were then quite excited about the fact. So we thought, okay, that seems like an interesting transcription factor. Can we actually go the next step and use this? And we were quite excited to see that we could actually target TCF4 pharmacologically, not directly, but indirectly. And this was done by work from Michael Green at the MD Anderson. In lymphoma, a special form of lymphoma, they used BET inhibitors and could show that BET inhibitors were working on TCF4 expression because they had a lymphoma with a genetic amplification of TCF4, and it was highly sensitive to the BET inhibitor. So this is not specific to TCF4, but TCF4 goes down. So what we did first is we looked at open chromatin, performed ATAC sequencing on mesenchymal-like cells and on melanocytic cells. And as you can see here, this is the start, so the gene goes this way. This is TCF4, this is the promoter region, or even the enhancer region. And you can see that chromatin is largely accessible in mesenchymal-like cells, but not in the melanocytic ones. And this is scale. And more importantly, the BID4 bound traditional enhancer that regulates TCF4 was diminished once we challenged these cells with the BET inhibitor. Okay, this is the way we think this BET inhibition works at TCF4. And if you look then at gene expression with qPCR, this is a nice dose-dependent effect. I'm not going to go there. Just to show you, then we performed also RNA sequencing again in this mesenchymal-like cell line upon BET inhibition. And this somehow, this is exactly the same heat map, exactly the same genes. This nicely mimicked the silencing with the siRNA. So, yeah, I mean, mimicked, phenocopied, you name it, whatever you want. Right. So basically, I want to leave you. So what we're doing now is we're doing, of course, this in a mouse model. So we tried it in the NRAS model. It didn't really work. Now we're using a B16 model. I can show you in a minute why. And want to basically propose a combination therapy of BET inhibition together with anti-PD-1. But I want to leave you with this this little summary. So we believe that TCF4 in this case is indeed controlling the mesenchymal-like cell state. It's not doing it alone. As you saw before, there are other transcription factors assuring the cell state. PX1 is one of them, ZEB1 would be one of them, SOX9, and so on. But more importantly, TCF4 is doing not only that, assuring the mesenchymal-like phenotype, but it's also repressing this MRTF program and thereby acting on, repressing the melanocytic antigens and presentation. It's also negatively regulating NRC5, and I didn't show you anything about that, but it's promoting the mesenchymal-like phenotype and is basically promoting immune escape and resistance. This is the way we believe this is working. Just one summary slide. So I think, single cell is not the holy grail, but I think, having these system approaches or ecosystem approaches can still, bring us quite some way to think about the next steps, how to fight cancer. And I think my lab now, as a continuum, will exactly do that. So we will continue focusing on the ecosystem, using computational biology, a lot of, single cell omics, and also at spatial resolution to basically find new cancer biology. We will probably continue doing melanoma to some extent, but now at the Institute now I'm joining, there are other cancer entities where we're going to translate our knowledge. It's the summary slide. I probably don't have to read everything again, but we believe that melanoma growth indeed is hierarchically organized, at least in the model we showed you. I showed you there are about 6 evolutionary conserved cell states, 6 to 7 short-term exposure of anti-PD-1 remodels the ecosystem. I showed you 2 meta programs very early on being associated with response or non-response. I showed you the true mesenchymal, thanks to single cell resolution, we can really now pinpoint down the true mesenchymal-like melanoma cell state. TCF4 is playing a role in orchestrating this transcription program, and we can use BET inhibitors to at least resensitize some of the melanomas that have this mesenchymal phenotype in there to anti-PD-1 checkpoint therapy. And this is the people I'd like to acknowledge. So this is the old lab. People who did the work, of course. So there's 3 people in particular. There's Dennis, PhD student who did a lot of the TCF4 work. I showed you Takis before. He was doing the beautiful mouse work. There's Ewout, also MD PhD student. He was on the clinical side. He did all the beautiful stainings. And postdoc Yara Pozniak, she did all the single cell analysis on the human side. Collaborators, of course, funding bodies, and thank you for your attention. When the visions around